Synthetic biology in general, but the synthesis of whole genomes for a synthetic cell, has benefited enormously over the last 20 years by the increase in both the number of bases that can be read, that is DNA sequencing, or reading, which is shown here in the red line, and also in DNA synthesis, which is shown here in the yellow line as DNA writing. So DNA writing means creating DNA in the laboratory, and DNA reading, shown here in the red line, means sequencing existing DNA. So you notice the very steep increase in the reading or sequencing of DNA that happened between about 2007 and about 2013. And that's largely based on what's been called next generation or next gen sequencing, exemplified, for example, by Illumina sequencing, which gives you large numbers of very short reads. They're very useful, especially for resequencing genomes, but have really taken over in terms of allowing us to do massive amounts of sequencing. And just for reference, they show here what's happened in terms of computer chips, which continue to go up, but slowing down. And also notice that in terms of the synthesis of DNA, we have not made much progress since about 2005. Also notice that the scale over here is a logarithmic scale. So we're looking at tenfold increases from here to here to here. So all of this has been important in terms of thinking about how to make a synthetic cell. Just as important as the ability to make more or sequence more DNA has been the concomitant change in price. In particular, if you look at the cost of sequencing over the last 20 or 30 years, it dropped very precipitously from about 2005 to about 2008 or 2009. It has held fairly steady since then, and that is largely because the next generation sequencing techniques that came into use starting from about 2010 to about 2013 are now in place, but the cost is not decreasing any further. In contrast, if we look at the cost of gene synthesis, that decreased until about 2008, but has held fairly constant, I would say, over the last 5 years. And the cost of oligos, which are short nucleotides, typically up to about 50 base pairs, came down somewhat until maybe 10 years or so ago, and has continued to decline very slightly, but by and large, at this point around 2015, the costs are fairly constant. But that sharp drop in the cost of sequencing and the drop in the cost of synthesis led to the feasibility of making DNA that could then be easily sequenced for a synthetic cell. In preparing for this lecture and in reading about the synthesis of DNA, I came across something I didn't know that I thought might interest you, so I'll just mention it briefly. And that is that when we send in a sequence that we want synthesized into a gene, there is biosafety that's involved in that, as you see here. And so there are several checks. One is a sequence check, and the second is a country check, and the third is a customer check. So they want to make sure that this is a legitimate sequence that is not something that is a toxin or something that could be used by terrorists. They want to know that it comes from a legitimate customer and that they can be reasonably sure that what is being made in a scientific laboratory and that it will not be used in any way that would be a biosafety threat to the country. So what I'm going to talk about today is the synthesis of a new or a synthetic cell, and this, to date, for the synthesis of an entire cell, has been based on these free-living bacteria called mycoplasma. And the reason that they've been used is that they're very small. So mycoplasma are a genus that causes diseases both in humans and in animals, although there are also many non-infectious strains. But the key here is that they have very small genomes. So Mycoplasma genitalium is about 580,000 base pairs. Capricola is a little over a megabase, as is Mycoides. So we'll be looking at these three strains, and the first one, because it was the smallest that was synthesized, was the Mycoplasma genitalium strain. So even while scientists were thinking of how to synthesize a whole synthetic genome, they also had to think about the problem of how they were going to get a host cell to put it into. So having a whole genome in a test tube is one thing, but having it actually be able to enter and replicate in a host cell is another entire problem. So the first article that's relevant to that came out in Science in August of 2007. And I'll mention right here that virtually all of this work has been done at the J. Craig Venter Institute. So you'll see his name frequently. He does not do the experiments himself, but he initiated and has continued to find support for the many scientists at the Venter Institute that are really working on the forefront of synthetic biology. So the host in this case, or the final organism that they were going to change, was Mycoplasma capricolum. So this was the recipient, and Mycoplasma mycoides was the donor. Now in this case, we're not actually talking about any synthetic DNA. So in this case, they isolated the genome of Mycoplasma mycoides and modified it by putting in an antibiotic resistance gene, and were able to mark it, and we'll talk a little bit later about marking genomes so that you can then later establish that they are the synthetic one that you put in, although in this case it was not synthetic, it was just a marked genome. And they were able to transfer, or in the case of synthetic biology, they typically call it transplantation. They were able to transplant the Mycoplasma mycoides genome into Mycoplasma capricolum. So you recall that if you have an antibiotic resistance gene in a plasmid or in any piece of DNA, it gives you the power to select for that. And in this case, by selecting the antibiotic resistance, in this case it was tetracycline resistance, that they had engineered in the Mycoplasma mycoides genome, then by selecting for tetracycline resistance in Mycoplasma capricolum, they were able to, over time, because of the selection for antibiotic resistance, show that Mycoplasma capricolum now had the genome of Mycoplasma mycoides. Another step in this process was creating bacterial strains that have genomes that have been cloned and engineered in yeast. And the reason that's important is that in synthetic genomes, they're too large to propagate in the intermediate stages in a bacterial host. So they must be propagated in yeast, which means that eventually in creating a synthetic genome, there was going to have to be a way to move it from the donor to the intermediate host, which is a yeast, and then to the final recipient, which in this case was Mycoplasma capricolum. So in this, again, Science article from 2009, also from the Venter lab, they were able to show that they could take the mycoides genome, mark it, put in tetracycline resistance, put it in the intermediate host of a yeast, replicate it there, and then move it to Mycoplasma capricolum.